Well, hello everybody. Welcome to this feature oral session 4. We have Scott Rich, who is a postdoctoral researcher, fellow at the Kremlin Brain Institute in Toronto, Western Hospital of Canada. His lines of work are related to synchronization in neuronal circuits and networks, in particular applied to epileptic seizures. So the talk of today would be lost neuronal heterogeneity in human epilepsy. Epilepsy is a fundamental principle unifying epileptic etiology. Thank you very much, Scott, for being here.
Awesome. Thank you very much for the honor of this featured presentation. I'm really excited to present this work, which is a very interdisciplinary effort that's across both myself, Homera Moradi-Chamé, who is an electrophysiologist extraordinaire, Jeremy Lefebvre, who is one of my co-supervisors and a more mathematically minded neuroscientist, and Taufik Valiente, who is another one of my supervisors and is a clinician scientist. And as you mentioned, the focus of this talk and of my work is epilepsy. And one of the reasons we're interested in this is that it is the most common serious neurological disorder in the world. For those who aren't familiar, epilepsy is characterized by recurrent seizures, seizures being a hyperactive and hypersynchronous brain state that start in a localized region of the brain and then propagate throughout the brain, engulfing the entirety of the brain. And this is what leads to the debilitating physical symptoms of seizures that you're probably familiar with. One of the things that's very interesting about this disease is that it's been characterized going back to quite literally the dawn of modern medicine with the ancient Greeks and Hippocrates. And partially because of that, we've had fairly effective pharmacological interventions for this disease dating back more than a century. But what's interesting about these pharmacological treatments is that there seems to be a pretty hard ceiling for how efficacious they are. They only tend to work for about 2/3 of patients with epilepsy. The remainder are classified as having medically refractory epilepsy. So when we dig into these pharmacological treatments, what we find is that they arise from this hypothesis that seizures arise from excess excitation. This makes intuitive sense, considering that seizure is a hyperactive brain state. So these pharmacological treatments tend to act in one of two ways, either by increasing inhibitory signaling via the neurotransmitter GABA, or decreasing excitatory signaling by blocking the neurotransmitter glutamate. But again, the fact that there's this hard ceiling of the efficacy of these pharmacological interventions implies that this hypothesis doesn't encompass the entirety of epilepsy. It doesn't encompass the entirety of all the different pathways to seizures. And if we want to design new pharmacological or other treatments for these medically refractory patients, we need new hypotheses underlying both epilepsy, the disease, and seizure, the phenomenon. Now, one of the features of seizures that is less classical, but has been the focus of more recent research, particularly computational research, is that a seizure is an information-poor state. This hypersynchronous activity is poor for information coding. Now, in parallel to that, there's been developing literature showing that biophysical heterogeneity is a preserved feature throughout the body, throughout biophysical systems, but specifically in the brain as well. And in the brain, it's known to promote information coding. So the fact that heterogeneity promotes information coding and seizure is an information-poor state implies that there may be some relationship between the onset of seizure and the loss of biophysical heterogeneity. Now, there's a wide range of sources of biophysical heterogeneity in the brain. There's diversity in neuronal cell types. There's been a ton of great posters at this conference about the diversity in interneurons. There's also diversity within similarly classified cell types in terms of biophysical properties. And then finally, there's always diversity in the inputs to particular units in any neural circuit or neural system, given the relative sparseness of synaptic connectivity. What we realized when we tried to sort of piece these two things together is that the seemingly disparate causes of epilepsy can actually be recontextualized as losses in biophysical heterogeneity. So for instance, certain types of epilepsy come about due to the loss of specific cell types. The SOM somatostatin expressing interneurons are implicated in this. There are certain types of epilepsies that arise due to the selective loss of these neurons. There's also certain types of epilepsies that come about due to the misexpression of ion channels, which can yield homogenization of intrinsic properties of neurons. For me, the most illustrative example of this is the H channel, something that's been talked a lot about in this conference. Something that's really interesting in epilepsy is that both the underexpression and overexpression of the H channel can lead to different types of epilepsies. And while this may seem contradictory, you can reconcile this, this contradiction through the idea that both this over and underexpression can homogenize intrinsic properties in these neurons. Finally, it's known that seizures, particularly those brought about by traumatic brain injury, are associated with synaptic sprouting. The additional synapses brought about by synaptic sprouting can be thought of as homogenizing the inputs to this system. So taken together, this allows us to hypothesize that epileptogenesis can be recontextualized as a process where a reduction in biophysical heterogeneity renders neural circuits less resilient to seizure onset. And as I mentioned, there is, bits and pieces of evidence in the existing literature that can be contextualized as support for this hypothesis, but it's something that's never been directly studied. And because it's never been directly studied, we wanted to approach this problem using a wide range of interdisciplinary tools. And this sort of experimental computational theoretical cycle, which is something that's motivated a lot of the research that we've highlighted at this conference so far, is how I'm going to organize the presentation of this talk. We're going to start with the biophysical reality, the experimental reality. I'm very lucky that I have access to data taken from live human cortical tissue. This is facilitated by Dr. Valiente's status as a clinician scientist. He is the one performing resective surgeries for patients with epilepsy. He directly takes this live cortical tissue. It goes directly to the lab where Dr. Muradi Chameh is able to do patch clamp recordings on these individual neurons. It's worth emphasizing that we're really lucky at Krembil and Toronto Western Hospital to have access to this data. Very few labs in the world are able to do recordings from individual human neurons with this level of precision. This unique data then constrains my computational modeling. I'll go into the details of our network later on. In turn, we're able to further probe these networks using tools from mathematical analysis. This is something where my other supervisor, Dr. Lefebvre's expertise as a more mathematically minded neuroscientist is particularly useful. And then finally, I'm going to conclude my talk by sort of completing this loop by showing how we can actually explain some very surprising experimental results using the theory that we generate in this process. Now, to start off with this experimental ground truth, the background to this research is our characterization of, quote, seemingly normal human cortical neurons. I use that terminology, seemingly normal, very purposefully. All of this tissue is taken from patients with epilepsy. The reason being obvious ethical concerns. We can only do surgery on humans when there is a medical need to do so. It is the convention in the field of human electrophysiology that tissue taken from the non-epileptogenic region far away from the seizure-generating zone is deemed healthy enough or seemingly normal to be classified and studied as indicative of the healthy brain. And we recently published a study in Nature Communications that goes into these properties of pyramidal cells throughout the cortex. One of the interesting things we found in this study was notable heterogeneity in passive properties, even amongst similarly classified neurons. So if you look at these layer 5 cells, there's about a 30 millivolt spread in their resting membrane potential. This is something that was somewhat surprising, especially relative to the existing rodent literature, but it's actually borne out by the other electrophysiological studies of human cortical neurons that exist. And I'll mention here that as a sort of spin out of this huge project, we were also able to develop a multi-compartment biophysically detailed model of a human cortical pyramidal neuron with a specific focus on the H current. And we identified how the H current is actually very different in human neurons as opposed to rodent neurons. If you're at all interested in using a more human-motivated model in your research, this might be a great place to start. Now, out of this study, in our characterization of these seemingly normal neurons, we wanted to compare this to unhealthy neurons that are more indicative of the situation of patients with epilepsy. To do so, we looked at 3 different sources of pyramidal neurons. The first is non-epileptogenic temporal lobe tissue taken from patients with epilepsy. This is the type of tissue that we use in this Nature Communications paper. We contrast that with epileptogenic tissue taken from the frontal lobe. This is epileptogenic tissue because it's taken directly from the seizure-generating zone that's identified via prior to surgery. And then finally, we had an additional control where we looked at non-epileptogenic tissue from the frontal lobe, this time from patients undergoing tumor resection rather than undergoing surgery for epilepsy. And again, we use this as a control to show that the results we find are not confounded by the different brain regions in which we're able to obtain tissue from our patients with epilepsy. Now, what do we find when we experimentally characterize this tissue? Well, we're primarily interested in excitability, considering that epilepsy is thought as a disease of excitability. And so we characterized a very simple distance to threshold measure, which is just the neuron's threshold potential and resting membrane potential subtracted. And what we found in our 3 different regions is a significantly lower coefficient of variation in this distance of threshold measure in our epileptogenic population when compared to either of our non-epileptogenic populations. This shows that there is a significant decrease in variability or heterogeneity in this measure in this epileptogenic population, and that this isn't confounded by the cortical region, considering we still see this difference when comparing frontal epileptogenic tissue from frontal tissue taken from tumor patients. And these differences can probably be more easily appreciated in these Gaussian fits to the data. There's this higher peak in the red epileptogenic curve. Now moving forward, when I start talking about our math and our models, I'm going to start talking about sigma, which is the standard deviation in this distance of threshold measurement, sigma E referring to our excitatory pyramidal neurons. When we look at just the standard deviation of these distance of threshold values, we find that they are significantly different between our frontal epileptogenic and temporal non-epileptogenic regions. So these are the values for either unhealthy epileptogenic low heterogeneity versus healthy non-epileptogenic high heterogeneity cases that we're going to use throughout the remainder of this work. Now, we wanted to create a mathematical computational system in which we could directly implement and control these heterogeneity values. We created a spiking EI network of Poisson neurons in which we could control this heterogeneity. For those who aren't familiar, these Poisson neurons are a stochastic process in which you implement a activation function. In this case, our activation function is of a sigmoidal shape. And then at each time step, we use a Poisson process to determine whether a spike occurs or not based upon the sort of input probability generated by this function f. Now, part of this function f is this term h, which we call the Rio base. Note that this is not the biophysical Rio base. This is just a sort of artifact of some differences between the mathematical modeling terminology and the electrophysiological terminology. This is just sort of a shift in terms of this activation function. And it's analogous to the distance to threshold. And it's also normally distributed with a standard deviation of either sigma E or sigma I, depending upon the population. So you can see here how our Rio-based distributions for our high heterogeneity case are wider. For the low heterogeneity case, it's much narrower. And what this HJ term does is shift these fainter sigmoids left and right. These fainter sigmoids in the background are the individual neuron sigmoids. The bolded sigmoids are population averages, which we'll talk more about later in the talk. I'll also mention this gray box later in the talk. It is there for a reason. It's a little bit of foreshadowing here on my part. But again, the idea here is that larger values of HJ shift these sigmoids to the right, essentially meaning that you need more input in order for the corresponding neuron to spike. That's akin to having a higher than average distance to threshold. And correspondingly, when these HJs are low, you're shifting the curve to the left, you're requiring that the neuron requires less input in order to spike. That's akin to having a lower than average distance to threshold. So you can think of this Rio-base as basically just a mapping of our distance to threshold, where we take our average distance to threshold and sort of map it to 0. So we have this sort of classical normal distribution that makes more sense for future mathematical analysis. Now, this is an EI network, so we have both inter and intra connectivity between our excitatory and inhibitory populations. A couple of things of note here is that there are differences between these two populations, both on their time constants as implied by experimental literature, as well as in the tonic bias current that sort of biases their baseline activity levels. And I'll finally note that there is noise in the system. There's an independent noisy input to each neuron. This noise is scaled endogenous to the system dynamics. So while it's there and it's participating, it isn't driving network dynamics. This is not a noise-driven system. I'll note our synaptic weights. As you might expect from an EI network, the EI and IE connectivities are much stronger than the EE and II connectivities. If you've seen a Poisson neuron before, you might look at these synaptic weights and say, whoa, those are really large. The reason they're really large is this choice of our nonlinear gain term, beta, which affects our activation function little f, that sigmoid. We chose this value of beta so that the heterogeneity levels made sense with millivolt scale, so that we could directly implement a sigma of 4.4 millivolts or 7.8 millivolts and yield dynamics that make sense. So again, this is basically just a rescaling of a more traditional Poisson neuron, and that rescaling has to be reflected in all of the other terms, including our synaptic weights. And for more details on that process, you can read the methods section on our bioRxiv paper. Now, what do we see when we start investigating these networks? So what I'm showing you here first is the case of a healthy system that has both high levels of excitatory and inhibitory heterogeneity. You're seeing a raster plot in the background here. In the foreground, you have quantifications of the network dynamics in 500 millisecond moving time windows, a synchrony measure here in blue, a measure of the average excitatory firing rate in black, and the average inhibitory firing rate in gray. And what we're doing, our sort of default experimental setting, is a slowly increasing drive to the system. And what we see is that all of these measures seem to scale linearly with the drive to the system. There aren't many major notable changes in network dynamics. Contrast that to the case where we have low heterogeneity, both in our excitatory and inhibitory populations. Now we see this very sudden onset of very synchronous dynamics that corresponds with a sudden uptick in the excitability of excitatory and inhibitory neurons. This is what we deem as the onset of seizure-like activity. Note that I'm focusing on seizure onset. We're not making any claims about whether this models a seizure. That sort of goes beyond the scope of this work and the sort of the simple Poisson neuron model that we chose for future mathematical analysis. We're focusing on the onset of seizure-like activity. And again, because seizure is a hyperactive, hypersynchronous state, this is a reasonable approximation for that onset. Now, obviously, I'm not going to convince you of anything by showing you plots from one run of these networks. So let's look at some averages. So now we're taking our measures and averaging them over 10 independent simulations. These fainter curves represent plus or minus 1 standard deviation. What you see in the high heterogeneity case is that this behavior is still very stereotyped. We see this sort of linear increase in the synchrony as a function of the external drive. If you were to think about what's going on from a population level behavior measure, something akin roughly to an LFP, you would see this, not many changes in the dynamics of the network beyond maybe a slight increase in activity. Contrast that again with the low heterogeneity case, where we see this very sudden uptick in synchrony and activity. We note that this, the timing at which, or the drive level, excuse me, at which this uptick occurs is again highly stereotyped. You can see how tight this standard deviation window is here. And what this would look like in some sort of population level measure is the sudden onset of high amplitude oscillatory dynamics. Now, what I've mentioned here is if you take a very critical eye, probably at worst anecdotal evidence that this lost heterogeneity makes the system more vulnerable to seizure onset. If I want to strengthen this and make a potential causal argument, this is where the mathematical analysis comes into play. So we're going to take our spiking network and reduce it via mean field analysis. I won't go into the major details here, but it follows along the lines of recent work by myself and Dr. Lefebvre. And what this yields is a much simpler two-dimensional system of two ODEs, one term measuring the mean excitatory activity, one the mean inhibitory activity. The main difference here is that instead of considering little f's for each individual neuron, we're now considering big F's, which are the averaged activation function over the entire population. So again, this is essentially just averaging together all of these background sigmoids into these bolder sigmoids in the foreground. The major difference being this blue curve, which is the high heterogeneity curve, looks flatter. It looks a little bit more linear towards the middle, in contrast to the much clearer S sigmoid shape for the low heterogeneity curve. Again, foreshadowing a bit here, that's going to be very important towards the end of this talk. With these tools in hand, we're able to go to town with our dynamical systems analysis. We can find fixed points. We can then linearize the system and find the stability of the fixed points by finding the eigenvalues of the linearized system. And what we're going to do is do this numerically. Because the fact that F is a convolution, it makes finding an analytical solution quite difficult. So we solve these things numerically. And we're going to do this for each level of this external drive that we looked at in our spiking networks previously. So we're going to maintain that correspondence between our spiking network simulations and our mathematical analysis. Now, what we find is that in the high heterogeneity case, we only ever get a single fixed point, which we can classify as a stable oscillator, considering it has an imaginary eigenvalue with negative real part. The fact that we have a single type of fixed point makes sense, considering we don't see any major changes in network behavior. Again, this is highly contrasted with what we see with low heterogeneity. First of all, we see multiple fixed points for low values of the external drive. The saddle and the sink collide and disappear, leaving only the unstable oscillator, a fixed point with, again, a imaginary eigenvalue, now a positive real component. This colliding of the saddle and the sink indicates that we're undergoing a saddle node bifurcation. And what's really interesting is that the value of the drive at which this saddle node bifurcation occurs corresponds almost identically with where this transition into hypersynchronous behavior happens in our spiking networks. So this clear correspondence between the mathematical system and the computational simulation allows us to strengthen this causal argument. It allows us to say that decreasing the heterogeneity leads to the saddle node bifurcation. Again, the only thing we changed in these analyses were the values of sigma E and sigma I. The existence of that saddle node bifurcation seems to be causal for this sudden change in synchrony that we deem our onset of seizure-like activity. Now, we wanted to see that this was more robust to varying values of sigma E and sigma I. For our spiking networks, we created what we called a bifurcation measure. Essentially, what this measure does is detect whether sudden changes in synchrony occurred by smoothing the synchrony measure time series, calculating the difference quotient at each time step, and then calculating the variance of these difference quotients. In the case where this is linear, you're basically going to see very similar difference quotients and low variance. When there are multiple components of this time series, you're going to see a multimodal distribution of those difference quotients leading to higher variance. Now, this bold border you see on the heat map demarcates systems that are multi-stable from those that are have single fixed points in our mathematical system. So again, this bottom left regime is multi-stable. The remainder of the heat map has a single fixed point. So what you can appreciate from this visualization is that there's this asymmetric effect of sigma I versus sigma E on both the bifurcation measure and on where this multi-stability occurs. This is a little bit more easily appreciable when we look at a slightly simpler experimental setup. Now we just give a tonic drive to the system, measure the network synchrony, and then look at whether the fixed point is either unstable in this bottom left or stable in the remainder. And again, you see this really nice correspondence between the regime of the highest synchrony, indicative of epileptic-like behavior, and this regime of unstable fixed points, which we saw in the low heterogeneity exemplar case. This asymmetry makes some sort of sense when you consider that there is, in all likelihood, higher heterogeneity amongst the population of interneurons, considering how diverse the subpopulations of interneurons are. So what this suggests is, one, that neural circuits can probably tolerate less lost heterogeneity amongst inhibitory cells as opposed to excitatory cells, the fact that this transition occurs at a higher value of sigma I, and also that preserving inhibitory heterogeneity might be a bulwark against the effects of lost excitatory heterogeneity. The fact that at this high value of sigma I, you always see very low synchrony. So taken together, this is support for our hypothesis that epilepsy can be recontextualized as a progressive loss of biophysical heterogeneity. We've shown that this exists experimentally in human epilepsy. We've shown computationally that the experimentally seen changes in heterogeneity yield dynamical changes associated with seizure onset. And then we use mathematical analysis to give a fundamental explanation for how heterogeneity affects the dynamics in the system's underlying mathematical architecture. Now, here's the really cool sort of, kicker to the story that finishes off that experimental theoretical loop. When we characterized our neurons, we also looked at their excitability via an FI curve, repetitive firing frequency on the y-axis, tonic input current on the x-axis. We were really, really surprised to find that our epileptogenic neurons were less excitable than their non-epileptogenic counterparts, oftentimes significantly so. This is really counterintuitive when you remember that epilepsy is typically thought of as a disease of hyperexcitability, but our single neurons didn't seem to be hyperexcitable. This confused us for a long time until we took a look back at this mathematical characterization of our neural populations. And what we remembered is that these average FI curves, these bolded curves, show this type of dynamic. The healthy, non-epileptogenic, high heterogeneity population is more linear and shows this higher activity level early in the sigmoid, in the region that corresponds with the FI curve. And lo and behold, I can just plop that FI curve onto this curve and see this remarkable correspondence between the experimental data and the effects of heterogeneity that we characterize mathematically. To strengthen this, we show this experimental data directly against that portion of the curve, that gray box. We also performed the best fit to this data, and we found really nice correspondence between the values of sigma that give us the non-epileptogenic and the epileptogenic data. What this shows is that the linearizing effect of heterogeneity explains the counterintuitive hypoexcitability of our epileptogenic, less heterogeneous population. And what's interesting about this is that it leads us to the idea that increased network excitability, a la the type we see in seizure, can potentially arise independently of hyperexcitability of individual neurons. We can get hyperexcitable network dynamics without hyperexcitable single unit dynamics. In fact, counterintuitively, less excitable single unit dynamics can lead to hyperexcitable network dynamics. So this is a really cool result that sort of highlights the complexity and the nonlinearity of neural networks, which is the reason why we're all here and doing computational neuroscience. Now, the end goal of this work and all of our work in the lab is to help patients with epilepsy and potentially uncover new treatment regimes or paradigms for these medically refractory patients. Part of our ongoing work is trying to separate out the effects of heterogeneity and the effects of this FI curve. We're doing so by creating new neural models that are a little bit more biophysically detailed, through which we can disentangle these influences. We also want to translate our findings to the clinic, and we potentially design ways in which we can add heterogeneity back to the system. This was the subject of a theoretical paper that we recently published in Scientific Reports. And then finally, we think that one way that which we could do this is the use of closed-loop neuromodulation and neurostimulation. And a really exciting new element of our work is that we've spun out a company called NerveX from the Valiente lab that's specifically targeted at deploying these implantable devices to design new treatments for epilepsy. And this is driven, of course, by Dr. Valiente, as well as Gerardo O'Leary, who is a soon-to-be PhD from the electrical engineering side of the lab. So with that, I want to say thank you again for the opportunity to present this work. I want to thank Drs. Valiente and Lefebvre, also Dr. Skinner, who is another one of my supervisors, not involved with this work as much, but I'm really lucky to have 3 supervisors who have a diverse range of interdisciplinary skill sets. Dr. Muradi Chanmay is, I would argue, the greatest patch clamp electrophysiologist in the world. I'm so lucky to get to work with her and get to use her amazing data. And then finally, our funding sources. And obviously, I will take all your questions now, but I'll mention that I'll be active on the Discord channel, FO4. And then you can also reach out to me via Twitter if you have any questions or want to discuss this work any further.
Okay, Scott, thank you very much for that interesting talk. Very, very interesting results. We have some questions. I'm not sure if we have time for all of them, but let's try to start. The first question is, does changing the ratio of excitatory to inhibitory neuron alter the importance of inhibitory variability?
So that's an interesting question. I do not believe so. So there is work from another group at U Ottawa that has looked at this sort of asymmetry and basically shown, I believe that it's work from Andre Longton, and I believe that those papers use 500 equal populations of excitatory and inhibitory neurons, and they still characterize the sort of asymmetric effect of heterogeneity in these populations. Obviously, the focus of that work was in epilepsy. It was a little bit different, but I would strongly believe that wouldn't affect things. But I would also say that this ratio of excitatory to inhibitory neurons is something that's pretty well established is the biophysical reality. The sort of 4-to-1 ratio is the sort of go-to for these cortical EI networks. So I don't think that changing that would be something that we would want to do since we want to sort of maintain that direct connection to the biophysical reality.
Okay, thank you. The second question is that you can actually observe stable oscillation in the system, because you mentioned the combination of a pair of imaginary eigenvalues and the corresponding real negative eigenvalues. But wouldn't that be representative of a stable focus rather than a stable limit cycle solution?
Okay, so in terms of do we actually observe stable oscillations, the answer to that question is quite simply yes. We did a lot of simulations where instead of doing this increasing drive, we just gave a tonic input. And if you look at those networks, you can simulate them for long, long periods, and you're going to see very stable oscillatory activity. So that's the answer from the computational side. From the mathematical side, yeah, so you're right that if we have a stable oscillator, we're expecting that to sort of cycle down and end up at that fixed point in that sort of nice little spiral. The existence of noise in the system prevents that from happening when we sort of actually simulate these things. So yeah, so from a mathematical standpoint, you're right. And again, some of those noise elements sort of get averaged out in the mean field analysis. But the correspondence between the mean field analysis and our computational results indicate that yes, what we're characterizing actually cause oscillatory dynamics in the actual sort of spiking network setting.
Okay, thank you. The next question says, have you thought about using Fitzhugh-Nagumo model instead of the Wilson-Corwin model so that more interesting dynamics can be found in the spiking network that allows you to do bifurcation analysis without oversimplification?
So no, we haven't thought about that. That would definitely be an interesting idea. The motivation behind our use of Wilson-Cowan style models was the sort of preservation of the spiking results with the mathematical results. We wanted to maintain this direct connection, and we did that because the sort of Poisson neuron setting with this sigmoidal activation function is something that allows us to very directly simplify and do analysis via the Wilson-Cowan. So doing this type of study in the Fitzhugh-Nagumo model would be very interesting. Again, but that's sort of not something that we thought about directly, just because of what our sort of motivation was in maintaining this relationship between the math and the spiking network.
Okay. I don't understand the next question, but I'm sure you do. What about bifurcation diagram on the time constant?
So I'm assuming this is asking what would happen if we varied the time constant of the model. So we did not do that. Again, that is we set that, and we set that because of experimental data. So that was something, again, that we didn't vary because there was experimental data that constrained our choice of that time constant. I can't really speculate as to what that would yield. I would imagine it would yield something interesting, but that was sort of because we did have the biophysical constraint motivating that. That wasn't something that we played with. As we add more complexity to this model, something that we're interested in doing is adding in, different interneuron types to a similar type of model. Then we would probably have interneurons with different time constants, and maybe we would get into that with a little bit more detail.
Okay. The next question is related to the analysis of the model. This is from a theoretical point of view. Why is a saddle point bifurcation lead to a sudden increase in synchrony?
So this is a great question. So if this was a more mathematically minded talk, I would have gone more into the bifurcation analysis type stuff. So apologies there. But it's well established in the mathematical literature that saddle node bifurcations are basically associated with the onset of synchronous dynamics. So this I can't really answer this satisfactorily in 30 seconds or a minute. But I would say if you look at some dynamical systems textbooks or stuff online, I think there's actually a really good Wikipedia page on saddle node bifurcations. But basically, this is a known quality of this bifurcation mathematically. That saddle node bifurcations yield sudden changes in dynamics that typically are associated with oscillatory activity. If you look back at some other previous modeling work in epilepsy, like the Epileptor from Victor Jersa's group, they looked a lot at these bifurcations and how different bifurcations can correspond with different phases of sort of epilepsy, the onset, the offset of seizures. And what they basically found was that a saddle node bifurcation best explains the onset of hypersynchronous hyperactive dynamics that corresponds with a seizure. So that is something that there's a lot of literature on, but it's pretty well established from a mathematical theoretical perspective that there's this correspondence between a saddle node bifurcation and a strong uptick in synchrony.
Okay, we have two more questions. I think we can finish. That it looks like the work doesn't consider the structure of the network, like feed-forward or recurrent structure. Will those also part of the feature that will affect the dynamic of the problem?
Oh, okay. So the structure of the network wasn't the focus of our work, but it is a traditional EI network. So it is all-to-all connected. I should have mentioned that. But there is recurrent connectivity, right? The E cells connect to the I cells, and the I cells connect to the E cells. Unless the reference here is to recurrence structure more in terms of a machine learning or artificial neural network style setting. But in terms of an EI network, you're right that this recurrent connectivity is implicated in producing oscillations. A lot of my previous work was looking at ping rhythms, and ping rhythms are basically driven by this E to I to E connectivity. But in terms of this question, that is there. That is a major part of these network dynamics, right? There's a reason that our E to I and I to E connectivities are our strongest connectivities. I would posit, based upon how robust this is, that if we weaken the connectivity and made things more sparse, if we took away this all-to-all assumption, we would still see these dynamics. And again, the fact that there is this mathematical principle underlying our network structure is pretty strong evidence that this is a more generalizable, robust dynamic that would, hold up to some of these reductions in our assumptions, like the all-to-all connectivity.
Okay. Well, just a very quick, if you can respond for the last question, is whether you consider to include the different inhibitory cells.
Yeah. So I mentioned this previously. Yes. So the answer to that is yes. That is something that is a major focus of our sort of next steps on this type of research. It was a conscious choice to only use one inhibitory population here, because that facilitates the reduction of the system and makes the mathematical analysis more amenable to numerical study. The fact that we're only looking at a 2-population model. But yes, this is something that Alexandra Guet-McCrae gave a great talk yesterday, showing his cortical network with 4 cell types, with the VIP cells, the SOM cells, and the PV cells. Our interneurons were basically PV cell-like, something that we're looking forward to doing. And since Alex is a former member of our lab, and the Hay group is a collaborator of ours, we're hoping to sort of start to integrate our stuff together and add in some of the biological complexity of those different interneuron types to this study of epilepsy.
Okay, Scott, thank you very much. We are a bit late already. Thank you very much for this very nice talk, and good luck with your study.
Thank you very much.
Thank you.